In this presentation, we will be reviewing the 5 protein subunits of the electron transport chain. These subunits are complex 1, 2, 3, and 4, as well as ATP synthase, also called complex 5. Here's an overview of the electron transport chain. Some important definitions to know. Complexes 1 through 4 are protein complexes that link redox reactions to create a protein gradient that results in the production of ATP. A redox reaction, as a reminder, is an oxidation-reduction reaction that involves the transfer of electrons between two species. NADH and FADH are two important molecules in the production of ATP in the electron transport chain. A proton pump is an integral membrane protein that creates a proton gradient across the cell membrane. Cytochrome molecules are redox-active proteins that contain a heme group, an essential iron atom that acts as a cofactor. And lastly, we have a heme group. Any heme group consists of an iron atom and a porphyrin. Heme groups are found in hemoglobin, a central component of our blood.
Structure of ETC. ETC is composed of 4 multi-protein complexes, complexes 1 through 4, that are embedded with inner mitochondrial membranes and have 2 small diffusionable electron carriers shuttling electrons between each protein complex. The history of ETC. ETC was discovered by Albert Lehninger at the University of Wisconsin in 1961, and he also found the 2 electron carriers in ETC.
The first protein complex in the electron transport chain is complex 1. Complex 1 is also known as NADH dehydrogenase, and it is a subunit within the electron transport chain. Complex 1 is located in the inner membrane of the mitochondria. It contains 46 polypeptide structures and is an L-shaped subunit. Here's the process of complex 1. In the first step of the electron transport chain, NADH, a coenzyme, binds to the protein. It is important to remember that the NADH used in this process is made through glycolysis and the citric acid cycle, also known as the Krebs cycle. NADH then transfers 2 high-energy electrons to a part of the enzyme, thereby oxidizing it to NAD+. These electrons then flow through cytochrome electron carriers, activating a mechanism that releases 4 protons into the intermembrane space. The electrons finally land on ubiquinone, a lipid-soluble electron carrier, represented by the letter Q, reducing it into ubiquinol, which diffuses within the membrane. Complex 1 then moves 4 protons across the intermembrane space, producing a proton gradient. This formula shows the oxidation of NADH and the reduction of ubiquinone into ubiquinol more clearly.
Also known as succinate reductase, complex 2 is the second complex of ETC. Location. Like complex 1, complex 2 is located in the inner membrane of the mitochondrial matrix. However, unlike complex 1, complex 2's function is not to be a proton pump, and it does not move protons into an inner membrane space. Complex 2 is composed of 2 hydrophilic proteins, an iron-sulfur protein and a flavoprotein, as well as 2 membrane proteins. The purpose of complex 2 is to convert succinate to ferminate with the help of the enzyme succinate dehydrogenase. 2 electrons are extracted from this FADH2 that is produced, and these 2 electrons travel through complex 2 and go through iron-sulfur clusters to reach the coenzyme ubiquinone. After reaching this ubiquinone, the 2 electrons turn the ubiquinone to ubiquinol. So, let's look at this through a diagram. As you can see, 2 FADs come together to form FADH2, and with the help of succinate dehydrogenase, the FADH2 produces 2 electrons. These 2 electrons then travel through complex 2 and through the iron-sulfur clusters, as can be seen here, and go to ubiquinone, which is turned into ubiquinol when the electrons reach it. Eventually, this ubiquinol goes to complex 3. So, what to know? What to know is that the main goal of complex 2 is is to convert succinate to ferminate and turn the ferminate and its electrons into ATP. However, less ATP is produced in complex 2 than the other 3 complexes, and it is important to remember that complex 2 is not a proton pump.
Complex 3 is the third protein subunit in the electron transport chain. Like complexes 1 and 2, it is located in the inner membrane of the mitochondria. It is composed of 3 major structures: cytochrome C1, which has 1 heme group, cytochrome B with 2 heme groups, and the risky center with a 2 iron, 2 sulfur group. The purpose of complex 3 is to transfer electrons from the ubiquinol produced in complex 2 and convert them into cytochrome C. This transfer is called the Q cycle. Here's the process of complex 3. First, the ubiquinol created in complex 2 binds to the protein subunit of complex 3. Remember, there are 2 protons and 2 electrons attached to this ubiquinol. The 2 protons are pumped into the intermembrane space, as shown in step 2. The 2 electrons follow 2 separate pathways, making up 1/2 cycle of the Q cycle. Here are the 2 pathways that the 2 electrons follow. The first one starts by binding to the 2 iron, 2 sulfur group of the risky center. Then it binds to the heme group of cytochrome C1, and then to the heme group of cytochrome C. Then the electron reduces cytochrome C and proceeds to complex 4. The path of the second electron is a little different. It first binds to the heme group of cytochrome B, and then is carried by a ubiquinone carrier to the second heme group of cytochrome B. Remember, in the structure, we mentioned that cytochrome B contains 2 heme groups. Then, this electron is reduced into semiquinone, which is an ion of ubiquinone. Here's a video that shows the 2 electron pathways that make up the Q cycle. This is the movement of electrons on the first pathway that makes up the half cycle. The electrons move from cytochrome C to complex 4, moving along in the electron transport chain. Here's the second pathway for the electrons, which move to the 2 heme groups of cytochrome B, and as shown, ubiquinone is converted into semiquinone by the electron.
All right, real run. If I mess up, well, if I mess up, you're not going to see this. All right. Complex 4, also known as cytochrome C oxidase, is the 4th major protein in the electron transport chain. It has 2 heme groups, A and A3, and 3 different copper atoms, both classified into copper A and copper B. Complex 4's main purpose is to transfer electrons from cytochrome C into oxygen to form water, pumping 4 protons into the intermembrane space. First, it oxidizes 2 cytochrome C molecules and stores the 2 released electrons in an iron atom in heme A3 and a copper atom. This allows 2 oxygen atoms from mitochondrial matrix to bind to them and to one another, forming a peroxide bridge between the iron and copper. Then, 2 more cytochrome C molecules are oxidized, and the electrons are sent to the aforementioned peroxide bridge to break it, binding to 2 protons from the mitochondrial matrix as well, to form hydroxides in the iron and the copper. These hydroxides immediately bind to more protons from the matrix to form water. Through the conformational changes of this enzyme during the process, 4 protons are transported into the intermembrane space. Unfortunately, though, the exact mechanics of this aren't very well known. Also, here's just a nice diagram of complex 4. It shows the exact process of what's going on in the journey that the electrons take through the enzyme.
ATP synthase. Sometimes referred to as complex 5, ATP synthase uses the large proton gradient that's already been established through the 4 previous complexes to release ATP and bring it to the mitochondrial matrix. There are 2 regions within ATP synthase, the F0 region and the F1 region. The F0 region is also known as the absorption domain, and the F1 domain is also known as the ring domain. F0 region receives these protons that have already been established through proton pumps and uses the high gradient in diffusion to bring them into the F0 region through the membrane. Then the F0 region will begin to spin, and the F1 region connected to the bottom of the F0 region also begins to spin. The F1 region is made up of all these different phosphate groups, and when these phosphate groups begin to spin, combined with ADP, ATP is formed, and that's how your cell gets its energy.
The electron transport chain is almost exactly the same in prokaryotes as it is in eukaryotes. However, instead of using the mitochondrial matrix, the prokaryote uses its cytoplasm, and instead of the intermembrane space in mitochondria, the prokaryote uses the space between its inner and outer membranes, also known as the periplasm.